Embryology is a fascinating field of study that delves into the intricacies of early human development. It explores the processes by which a single fertilized egg transforms into a complex organism, complete with functional systems and organs. One such system is the nervous system, which plays a vital role in coordinating and regulating our body's functions. In this script, we will embark on a comprehensive journey through the various stages and mechanisms involved in nervous system development. During the third week of embryonic development, a flat sheet of cells called the neural plate begins to form along the dorsal midline of the ectoderm. This process, known as neurulation, involves complex signaling interactions between multiple cell populations. The neural plate subsequently undergoes morphogenetic movements to transform into the neural tube, which serves as the foundation for the future brain and spinal cord. Neural crest cells are a unique population of cells that arise from the dorsal neural tube during neurulation. These cells possess remarkable migratory abilities and embark on extensive journeys throughout the developing embryo. Through a series of intricate molecular events, neural crest cells ultimately differentiate into various cell types, including sensory neurons, glial cells, autonomic ganglia, and pigment cells. Transcription factors, such as PAX6, ATX2, and SOX2, exert precise control over the differentiation of neural progenitor cells. These regulatory proteins regulate gene expression patterns, guiding cells towards specific neural lineages. Through the intricate interplay of transcription factors, neuronal and glial cell types with distinct functions and characteristics ultimately emerge. Axon guidance is a critical process that enables developing neurons to extend their axons towards their appropriate targets. Within the embryonic environment, a plethora of guidance cues, such as netrins, semaphorins, and efferents, provide directional signals to growing axons. At the leading edge of the axon, the growth cone, a highly dynamic structure, responds to these cues and steers axonal growth towards their intended destinations. Synaptogenesis marks the culmination of neuronal differentiation, as synapses are the functional connections between neurons. Pre- and postsynaptic differentiations occur, enabling the release and reception of neurotransmitters respectively. Various synaptic adhesion molecules and signaling molecules play crucial roles in establishing and refining synaptic connections. Furthermore, synaptic plasticity allows for modifications in the strength of connections and contributes to learning, memory, and nervous system refinement. In the peripheral nervous system, neural crest cells differentiate into sensory and autonomic ganglia. These clusters of neuronal cell bodies are critical for relaying sensory information and regulating autonomic functions. Signals from surrounding tissues and the concerted actions of various molecular pathways guide the aggregation, proliferation, and differentiation of neural crest cells into specific ganglia. Glia, once considered as merely supportive cells, now occupy a prominent position in neurodevelopmental research. Neurons and glial cells engage in bidirectional communication, ensuring proper migration, differentiation, and survival of both cell types. Glial cells, including astrocytes, oligodendrocytes, and microglia, provide structural and metabolic support to neurons and actively participate in the wiring and maturation of neural circuits. Glial cells, particularly oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system, are responsible for myelination. Myelin acts as an insulating sheath around axons, facilitating rapid conduction of nerve impulses. The development of glial cells is tightly regulated by a range of signaling molecules and transcription factors to ensure precise timing and appropriate myelination throughout the nervous system. The blood-brain barrier, a specialized endothelial interface between the blood and the brain, tightly regulates the exchange of substances between these two compartments. During development, endothelial cells of capillaries within the brain acquire unique characteristics and establish tight junctions, preventing the passage of large molecules and toxins while allowing the passage of essential nutrients. Understanding the mechanisms underlying blood-brain barrier formation is crucial for unraveling neurological disorders and developing drug delivery strategies. Neurotrophic factors, including nerve growth factor, brain-derived neurotrophic factor, and glial cell line-derived neurotrophic factor, play essential roles in orchestrating neurodevelopmental processes. These factors support neuronal survival, regulate axonal growth and guidance, and promote synapse formation and maturation. Imbalances in neurotrophic factor signaling can disrupt normal development and contribute to various neurodevelopmental disorders. Neurodevelopmental disorders encompass a range of conditions affecting cognition, behavior, and social interactions. Disorders like autism spectrum disorders and neural tube defects result from a complex interplay of genetic and environmental factors that disrupt normal nervous system development. Understanding the etiology and molecular mechanisms underlying these disorders allows for improved screening, diagnosis, and therapeutic interventions. The developing nervous system is vulnerable to the influences of the external environment. Exposure to toxic substances, medications, maternal factors like stress and diet, and infections can interfere with normal neurodevelopmental processes, with potentially lifelong consequences. Understanding the intricate interactions between genetics, environmental factors, and neurodevelopment provides crucial insights into preventative measures and the protection of vulnerable populations. In conclusion, embryology enables us to comprehend the intricate stages of nervous system development, highlighting the diverse factors involved. From neural tube formation to external influences, this knowledge enriches our understanding of human development.